My name is Aida Schwartz. I'm a medical geneticist based on Porto Alegre, South Brazil. And if we see in my background, I have a sentence in Portuguese, who means we are a reference service on rare diseases. And it's my pleasure to introduce you Professor Boycott, who will give us today, the lecture. Professor Boycott is a professor of pediatrics at the University of Ottawa in Canada, where she is a clinical geneticist at the Children's Hospital of Eastern Ontario, chair of the Department of Genetics, and a senior scientist at the CHEO Research Institute. Dr. Boycott is a Tier 1 Canada Research Chair in Rare Disease Precision Health, whose research program bridges clinical genomics to basic research and is focused on the understanding the molecular pathogenesis of rare diseases, aiming at improving patient care and family well-being. She leads the National Care for Rare Canada Consortium, integrating genomic and other omic technologies to improve our understanding of rare diseases, with a particular focus on solving the unsolved and most difficult rare diseases. To leverage these discoveries, she co-leads the Canadian Rare Diseases Model and Mechanisms Network, established to catalyze connections between newly discovered rare disease genes and basic scientists who can rapidly study them in model systems. Globally, she moves the rare disease agenda forward as part of the Global Commission to End the Diagnostic Odyssey for Children. And Dr. Boycott has more than 300 papers published in PubMed, so it's an honor for us to have you here.
Thank you very much, Ida. It's lovely to be here. Thank you very much for the invitation. And today I'm joining you with many perspectives probably on the complexities around rare genetic diseases. And I'm going to talk to you really about the cumulative knowledge from over a decade of work. And you can see the map of Canada there on the right. You can see the little red dots, and that is our clinical research network that has been connected since 2011 to try to address some of the challenges we see with rare genetic diseases. And just to start with, I want to make sure we recognize that February the 28th was Rare Disease Day. This is a day that the world recognizes since 2008, where we think about rare diseases, the individuals they affect and their families. Rare diseases are estimated to affect about 300 million people worldwide, but the types of rare diseases I'm going to speak to you about today are those that affect about 1 to 3% of the population, and they are those rare diseases caused by a monogenic or single gene condition. So you sometimes referred to as Mendelian. These are the rare diseases we're talking about here. And so the number is not as great as 300 million, but it's still very significant when you think about them collectively. We recognize right now in 2023, just over 6,000 rare genetic diseases or Mendelian diseases caused by just over 4,000 genes, but we estimate there are still thousands that are unsolved, which presents lots of challenges for families who are trying to find diagnoses within our systems. And this is the challenge I'm going to focus on today, and it's referred to as the diagnostic odyssey. And as you can see that line looping through, you can sort of just see some of the places where it loops, where it sort of says, you know, no diagnosis or the wrong treatment, wrong diagnosis, referral to specialists. It's a very linear process with some very much sometimes where it goes out onto its own sort of miss track, and then it comes again. And this process in and of itself can take anywhere between 5 to 8 years on average, and obviously very different depending on which part of the world you come from. But even in our Canadian healthcare system, our data suggests that it's 5 to 8 years for a typical patient with an undiagnosed rare genetic disease. And so I alluded to just a minute or two ago about the collaborative networks that we've been running in Canada that have helped to try to address some of the common challenges for rare diseases. And I will speak about, well, I'll mention 3 of them today, but I'll spend most of my time on Care for Rare. So Care for Rare initially began in 2011 as the Forge Canada project, but it is continuing and it is still running in present day. And it is focused on discovery of new rare disease genes and developing approaches to solve the most difficult rare diseases. And so the current 3rd iteration of this network is currently ongoing, and it's called Care for Rare Solve. And Care for Rare Solve is really focused on those disease mechanisms that are difficult to identify. I will also mention 2 other networks. The first is the RDMM network, and that's a Rare Disease Models and Mechanisms network. And I think many of the people joining us today will be interested in how we link to our Canadian model organism community to help sort of understand the mechanisms of some of these rare diseases. And I'll give you one example of that. And I will very briefly mention the All for One network, which is just launching now, which is really focused on data sharing and access to research for all Canadians, no matter where you live. And I will refer you, if you're interested in more information, to our publication just last year, which really described the outcomes of our gene discovery network over the past 10 years. So when we think about a diagnosis and the goal being a diagnosis for all rare genetic diseases, really there are 2 challenges with that. The first is access to the right expertise, to the right tests for families within a healthcare system. The second is making sure that we understand the mechanism of all those rare diseases so that we actually have the knowledge when you have sufficient access to be able to provide a diagnosis for these families. So I'm going to break those 2 apart. I'm going to start with access. And so I'm sure all of you have heard of clinical genome-wide sequencing. This is a term that I use to describe both exome and genome sequencing. So exome focusing on the 2% or so of the coding portion of the human genome. In most cases around the world, clinical genome-wide sequencing is still delivered as exome sequencing because it is still the most affordable route. And this is a patient of mine, Abby, and just you can sort of see this representing her diagnostic odyssey there on the left, where these are typical types of numbers that you're seeing here in terms of waiting for a diagnosis, receiving misdiagnoses, and seeing lots and lots of specialists. It's a very inefficient way to run a healthcare system. It's also a very expensive way to do it. And Abby, for example, has undergone at least $15,000 in diagnostic genetic tests. And unfortunately, she still is undiagnosed. Now, when we apply this test generally to patients within our clinics, the diagnostic yield for genome-wide sequencing is about in the 30% or say it's about 31 to 37%, depending really on the criteria and the eligibility criteria. But present day now, hundreds of thousands of rare disease patients have been clinically sequenced around the world. And in Canada, up until about 2 years ago, we had quite variable access to this testing. And largely that was because we didn't have the infrastructure set up within Canada to perform this test in province. And most of our testing was going out of the country through what we call an exceptional access program, where the testing was being done in mostly the United States. And so I became very interested in health services research to think about how to bring new technologies like clinical genome-wide sequencing into the healthcare system. And I sort of think about it in this way. The first is the top-down approach, where obviously new dollars come into a system and they roll a new technology down the hill. The national health system in England is a very good example of this approach. Many countries rearrange existing resources so without a new influx, but have a great deal of flexibility with respect to how they deliver testing. That's not us. I live in the bottom-up approach where you're trying to roll the rock up the hill to produce the evidence that the payers need to deem this a cost-effective test, and then reimburse it at scale. And as many of you will recall, we do have universal healthcare in Canada, though it is administered provincially. And so that essentially means we have 10 provinces and 3 territories who don't necessarily do or make the same decisions about reimbursement. However, once you have reimbursement, you are covering an entire region. So I am from the province of Ontario, and this is my real lived example of moving genome-wide sequencing into the clinic, which began in 2013 and came out of that maze in 2021. And I won't go into the details of all the things that we did, engaged with the various stakeholders across the healthcare system in terms of making decisions, but there was a lot to be done. And so there really are, it really is a difficult path to bring a technology in. And in most realities, it takes 10 to 15 years to bring a new technology into the healthcare system. But once it came in, and it was launched in 2021 as an in-province provided test called Genome-Wide Sequencing Ontario, the entire province who are eligible within our 15 million population now have access. And so once it's in, it is a fantastic way to provide equity. And I just want to make one note about bringing in this kind of new technology. And so it comes with a huge amount of data. And it's really important that it can't be re-siloed and kept from being part of a greater health data ecosystem, which would benefit all. And so our All for One health data ecosystem is a partnership we're putting together across the country that will allow us to share data for two purposes. The first was you want to make sure that no matter where you live in Canada, you have access to a high quality genome-wide sequencing test as standard of care. And the second is that we want to make sure that no matter where the family lives in Calgary, or in Canada, sorry, that they have access to precision health research for all Canadians. And so we've really come up with a two-pronged solution that we are in the midst of developing. And the first is called All for One Clinical. And what that is sharing variants across the entire country in the setting of a clinical diagnostic pipeline to ensure that the patients who are sequenced, for example, in Ontario, benefit the patients who are sequenced in Newfoundland, for example, and vice versa. And the second is giving patients who are undergoing this testing the chance to opt in to become part of a research registry, to connect with researchers who are potentially studying either their particular rare disease, or perhaps they're unsolved and they wish to undergo further research testing. But either way, all of this to think about making sure that no matter where you live, every patient has the option to participate in research if they wish. And the other thing I want to talk to you about, just very briefly about access, is the part where many of you may have heard that a lot of the diagnostic odyssey is because nobody ever thinks about a rare genetic disease, or they think about it quite late. And I just want to show you that AI is coming to play into this. And at my institution where I am in Ottawa, we have been on an electronic health record system for 8 years. It's called EPIC. And within that, we now have a huge data warehouse of information on all the hundreds of thousands of patients that have been seen in this hospital. And so what we are in the midst of developing now is an algorithm that would be used on the data in our system to flag patients who potentially may have a rare genetic disease and would be eligible for clinical genome-wide sequencing. And so the goal here is really to equip frontline providers with tools for both diagnosis and referral of appropriate patients. And so we are developing a rule-based expert system that will retrospectively identify patients with complex and undiagnosed rare genetic disease and flag them to their most responsible physician so that they can be considered considered for clinical genome-wide sequencing. And the idea in the future is that this would become something that happens at the beginning of care and not at the end of care and not in a retrospective fashion. And so you can sort of think about this Think Rare AI type of tool and its development over the next several years that would flag patients as they enter into a single door for care at our institution and similar institutions, with the goal that it can predict, with a high degree of confidence, patients that may have a suspected rare genetic disease, contact or flag within our electronic system the most responsible physician and say, have you considered a referral to genetics, or at some point, have you considered directly going to genome-wide sequencing, with the Department of Genetics actually then spending its time on the other end once the data is generated and seeing patients then for diagnosis and genetic counseling. So that's the vision that, as a clinician, I'm very interested in moving forward over the next 5 to 7 years or so. So that's really what when I think about access, you think about, can you get the appropriate tests funded in your system, and can you find the right patients to get the test in a timely fashion? But on the research end of things, I've been very focused over the last 10 years on the mechanism or the molecular pathogenesis of undiagnosed rare disease. And over that time, we've enrolled over 3,000 patients in our Canadian network to undergo additional data analysis and additional omic technologies to be able to start to uncover these very, very challenging rare diseases that cannot be diagnosed within our clinical system. And I really like this image where you can sort of see in the yellow, you've got this little girl in her pink dress. This is the diagnosis, but there are lots of reasons you may not get there. And I've put them in 4 big groups. And the first is that maybe the gene is not yet discovered. So as I mentioned, there's probably there's thousands of genes that have not yet been associated with human disease. It may be, though, that the genetic etiology of that particular rare disease is undetectable to the current technology. So this is where the little guy's hiding in the box. Or the etiology of that particular clinical presentation may be more complex or actually not genetic. And this is going to take much more time when we think about polygenic, oligogenic, polygenic, and gene environment interaction as huge probably contributors to some of the families that we see in clinic that we don't yet have tools to be able to sort out. But let's focus just right now first on the genes not yet discovered. And what is really important as we now start to generate more and more genomic genome-wide data in the healthcare system. And one of the reasons I'm so focused and interested in the All for One initiative in Canada is the secondary use of data for discovery. And it presents a new set of problems or challenges when this data is generated in the healthcare system, because all of those folks are data custodians that be set up differently in different jurisdictions, but there are data custodians in the healthcare system, and they don't like to give data away. So we need to sort out a way that we can facilitate that. And when you look at this apple tree, you can sort of see in the bottom green segment that these are the patients that are diagnosed by exome. So they're fairly straightforward. And in that clinical test, their diagnosis is there about 30% of the time. In the middle of the tree, though, are those patients where their disease mechanism may actually be sitting in that exome, but it is yet to be discovered. And then at the very top is where omic technologies need to come in, because their disease mechanism is outside the coding region, for example, or difficult to identify in the coding region. Now, each year in the online Mendelian Inheritance in Man, it reports 250 to 300 or so new disease gene relationships every year. So that means that, after 3 years, you have some upwards towards 1,000 new disease gene associations that one needs to think about. So it's an ever changing target, which means that your originally generated data is super important. But as I mentioned, as this data becomes generated by more and more different health data custodians. So for example, in the province of Ontario, where I live with 15 million people, there are 160 data health custodians. And that means it becomes siloed. So we need to find a way to leverage that, because there are discoveries to be made in it. And so what we've developed is a national data sharing platform called Genomics for RD. and it links all of our sites across the country, with the entry point being a software called Phenotypes. And Phenotypes allows us to describe patients using human phenotype ontology. Which means it's a computer readable way to describe patients. And in Genomics for RD, what we have done is set up a governance framework so that the patients are consented into this. This is a discovery platform. It's a research platform, not a clinical platform. And we can share it in a variety of ways, including open for very with data that's not identifiable, controlled for data that is, genomic data sets with associated phenotype for particular families. So it is got lots of information, so it's very protected, and then restricted, where we have ways that individuals can come in and access our system. Because they're trusted. And one of the main things that we're doing in Genomics for RD is something called matchmaking. And so matchmaking allows us to think about identifying new disease gene relationships in a number of different ways. So this is how I envision it. So the first, and I'll talk to you about that, is called two-sided matchmaking. And in two-sided matchmaking, there are two clinician scientists with a hypothesis that say, I think this gene causes, when you dysregulate it, causes this particular disease with this set of symptoms. So two people are looking for each other. It's like a game of Go Fish almost. And you come into this network to put that question into the network and find a match. One-sided matchmaking is when there's just one person who has this particular hypothesis and needs to come into your data set and have a look at it in a trusted way. And then there's zero-sided matching, which I won't talk about today because we're not very good at it yet. But it's where data sets just interact with each other. And eventually, machine learning and AI will help us make and uncover some of these discoveries. But let's talk about two-sided matchmaking first. So in Genomics for RD, if we have a candidate gene identified for a family, we put it into a node of the Matchmaker Exchange called Phenome Central. And Phenome Central is the Canadian node of the Matchmaker Exchange. And what the Matchmaker Exchange is a network that we have been running for the last 7 years that currently links 134,000 unsolved patients from across the world from 104 countries. So some of you on this call will probably be familiar with this. It currently has 15,000 genes tagged as somebody's being interested in matching somebody to somebody else who has an interest in that gene. And this has been a remarkable 7-year journey. And I think we're over close to 700 discoveries, so novel disease genes that have been identified this way. And I want to show you one example of this. And so this is Justin's story. You can see Justin here as a little baby when he was first seen at our institution. And here he is when we most recently saw him. He's now in his 20s. And Justin was unsolved after clinical testing. And his data was repatriated into the research side, and it was analyzed. And like Justin, about 20 to 30 patients, 20-30% of patients who have who are unsolved after their clinical testing, and you reanalyze their data, will have a candidate variant in a gene that's not yet been associated with disease that may be the answer for them. And in Justin's case, there were 2 novel candidate genes that were really interesting. They were both de novo, so occurred in Justin, not present in his parents, and we needed to find other people who had interest in the same gene. And so this is called two-sided matching. And so what you enter into the system is some, clinical description and the genotypic data, which in this case was the 2 genes that we were interested in for Justin. And it goes into the Matchmaker Exchange, and it finds you and sends emails that links you to other people in the world who are also interested in that gene. And you share information with those clinician researchers. And so in Justin's case, he had 2 genes we were interested in. The first one we matched with was USP7. And he had this is the first gene indicated here on the map by green. So those are all where all the patients lived around the world who also had de novo or new for them changes in that gene and had overlapping features with Justin. But we kind of paused on that because Justin had, an additional couple of features that the other patients that we learned about with USP7 didn't have. And as it turns out, 10 months later, the second gene we were interested in also matched. You see those by the indication in orange. That gene was associated with disease for the first time and actually explained the rest of Justin's phenotype. So Justin actually has 2 different rare diseases caused by de novo dominant mutations in his genome. So you can sort of get just a sense of the complexity of some of these young people who remain unsolved after clinical testing. So when we think about matchmaking, these are our outcomes. And so we have put 285 genes into the Matchmaker Exchange, and we have made discoveries for 53 of these. So 19% we have gone on and published novel disease gene relationships because of the Matchmaker Exchange. And that's just our Canadian local experience. It is still a laborious process. It has a high false positive rate. Most genes match the first time. And one of the major matchmakers in the system is called Gene Matcher, which is run from Johns Hopkins. And it really has engaged the community, but unfortunately it does not include phenotypic information, which means you got to exchange a lot of emails back and forth with other researchers around the world to be able to sort out whether or not that match is actually looking like it needs to be pursued. Either way, it is still like an amazing event, and obviously 53 discoveries is nothing to dismiss. Now, when we think about one-sided matching, I'm just going to show you just how we've set that up, is that we now have a portal that we're launching where anybody who's a trusted individual who we know is working on rare disease can come in and can go in and make a query on our system and say, do you have any, whatever the description of the variant is, in this particular gene associated with whatever phenotype, and the system will spit back out these answers. This allows this individual with the red puzzle to give a lot more have a lot more discretion on what they're searching. And our experience so far is that 3% of these queries result in a validated discovery that goes on to publication. And so this will expand and be taken up more broadly around the world. It puts more work on the individual coming with the query to make sure that they're winned. So I've talked to you about matchmaking, both two-sided and one-sided, in terms of trying to solve patients who are like Justin. But I want to just take a quick detour into the fact that sometimes model organisms can also play a really critical role in providing the functional evidence we need for causality, as well as functional and biological insights, set up for therapeutic screening, et cetera, et cetera. And so making these connections with this community is really important. And so around 2013, we had the idea in Canada that we needed to be able to increase collaborations between clinician scientists and model organism scientists that you can see here on the right for some of these model systems. And making those connections, but also when those connections are made, ensuring that there's a little bit of seed money for the model organism scientists to begin the experiments on the gene that us and other networks have discovered. And so this became the idea for what we call the Rare Disease Models and Mechanisms Network. Again, you can find more information on our webpage. And we set out in 2014 to launch here to accelerate and support these collaborations. And when matches are made and all the parties agree, the model organism scientists gets $25,000 for the first date, basically. And so it's kind of like a dating service, except there's a really, really, really great first date with $25,000 that goes with that, to do the first set of experiments, with the idea then that they may be able to take that preliminary data and go on to get a larger grant. And then you have brought in trainees and a new lab to discover a rare disease, of course, that is very important to several families in Canada, as you can imagine. So just to give you a quick example of this, and this is a new condition that we described in collaboration with a group from the US, which is just now under review at Nature Communications, where we had 2 families that had changes in the exact same amino acid residue in the gene MGP. And you can see a little photo here of this little girl, and you can see her lumbar lordosis. If you look at her x-ray, you can see the flattening of her spine. She's got, short tailored fingers, and there's some hip dysplasia, for example. And the two families had a very similar and overlapping phenotype and that very specific change. And so we went into the RDMM network looking for a match for MGP, and we made the great match with Professor Merched from McGill University in Canada, who was, turns out, an expert on MGP. And he developed a knock-in of this particular disease for us. And as you can see, and I won't go into all the amazing data that he has produced for us over the last 18 months or so, but you can see that the mutant mice are smaller, and if you look at them compared to controls, they have a very, very similar phenotype to the human phenotype, and so it's quite well recapitulated. So it's a fantastic way to make new friends in the model organism community, and then have somebody who's going to study a rare disease that's very important to a family, for example, that I'm following here from Ottawa. And so in the last 7 years, we have engaged 664 scientists in Canada who are part of this network that we can match to. And the other cool thing I'd like to point out is that we actually also have partnerships with over 15 patient organizations now who are interested in having model organisms for their particular diseases of interest. And we've been able to provide the scientific oversight for that to happen, as well as matching funding for them. And I think that's a great way to engage those communities, which we can definitely talk about a little bit more at the end of this talk if you guys are interested. All right, now I just want to move to what if the genetic etiology is not detectable to the technology that we were using. And so this is the little guy in the box. So we've gotten the data back, we've looked at the coding sequence, can't find it. So what do we do then? And so this really is where the next wave of technologies and how they're applied to unsolved rare disease patients comes into play. And so as we talked about, the exome sequencing is really great at nailing that coding sequence. But what about read, the long-read genome sequencing? We have sequencing of RNA, we have deep sequencing for mosaicism, and then we have metabolomics, lipidomics, and epigenomics really is secondary functional assays to sort of point you in the right direction or confirm that you're thinking about a mechanism that might make sense. Now, when exome sequencing came along in 2011, it was pretty straight road. It's like the railroad track just went down. It's pretty, you put the right families in there and you can solve a lot of rare disease. With multi-omics, it's more complicated. When do you use what for what family? Because all of them are quite expensive. And so you can't, unfortunately, at least where we're from, do everything for everyone. And so we developed a research protocol from a meeting that we held in 2018, and we published our first concept of this in 2019, and then again in 2020. And what you see on the right is actually quite overwhelming probably to look at, but I'm going to break it down just very high level for you, just to give you an idea of how we make decisions about what family gets what. And we thought of our unsolved cohort here on the left as in 4 groups. And then in the 4 groups, we apply the technologies just a little bit differently based on the hypothesis of where we think that underlying variation is, where is it hiding? And so let's just start with the first group. And the first group are those where it's a very specific clinical entity. For example, say neurofibromatosis type 1 or tuberous sclerosis or cystic fibrosis, where we should know that those are the key 1 or 2 genes that cause that phenotype, but we can't find the variant for our particular patient. And so this is really where this mutation is undetectable to the technology. We know where to look, but we can't see it. And so things like whole genome sequencing and RNA-seq are very important to figure out that mutational spectrum. The second group are those where there's a recognizable disorder, but there's no genetic test for that. This is one of the most interesting groups that remain, in the sense that there are very few recognizable clinical genetic syndromes that are left where we haven't discovered. There's literally about 10. And many of them are actually going to prove to have very, very interesting mechanisms, and we'll likely learn a lot from that. But many groups around the world, including us, have put exomes and genomes on these very well phenotyped patients and not been able to find them. So some of them are probably mosaic. Some of them are probably much more complicated interactions, which we don't yet quite understand. And so we will learn a lot about these things as we go forward. The third group is probably and the fourth group are the most common things that we see in the genetics clinic. They're, these heterogeneous groups of phenotypes where lots of genes can cause it, for example. So maybe the phenotype is recognized in group 3, whereas in group 4, it doesn't actually have a name. But you got to have data sharing with that. You have to have at least genomes and RNA-seq with that, and sometimes even long-read genome sequencing. And in the last group, the reanalysis of the existing data that might already be there for those patients is really, really important. So when you look at this protocol, everything in green is under a research protocol, and everything in gray, which includes things like sharing data through the Matchmaker Exchange, should be considered part of clinical care, in our opinion, because we should be taking those extra steps for our families to resolve both variants and genes of interest within the clinical setting and not necessarily leaving it to research anymore. So I want to show you a couple stories of how these multi-omics have worked. And so this is a little guy I've followed since he was 11 months old. His name is Colton, and you can just see a little bit about his diagnostic odyssey on this timeline here. And he was enrolled very early on in our first research project. And we sequenced him before you could get clinical exome sequencing, and had a candidate gene which we never could prove. We did some functional tests, but we never matched in the Matchmaker Exchange, which is a big kind of flag that maybe it's the wrong gene. And so eventually in 2021, he got research genome sequencing. And what that showed us is that this is Colton in yellow, and his mom and dad. Dad is in blue and mom is in pink. And what you can see is that Colton is missing 3 exons of a gene called TBCK, which we could not see on the exome, could not see on a chromosome microarray, but could see in the short read genome data. And his mom and dad are both carriers of the exact same exon deletion, despite being non-consanguineous. And so this explained Colton's presentation after 10 years, almost to the day that I saw him. And without the genome sequencing as the approach, we never would have been able to uncover that mechanism. And he actually fits very closely, very, very well with this group of kids who over the last 5 years have been recognized to have recessive variation in the gene TBCK, which causes, very significant hypotonia and global developmental delay. And this is Colton and his parents after their 10-year diagnostic odyssey. And, you can just read those words if they're of interest to you. But I think they just really highlight that it's really, for them, we can't treat Colton. We don't have a therapy for that particular rare disease. But it's really being able to connect with the communities through Facebook and other types of patient organizations that have allowed them to not be, all by themselves anymore. And now I want to show you something about the next technology. And so this is Austin, who I've also known for more than 10 years. I just saw him this week, actually, and we decided that I've known him for 15 years. And he's been unsolved since that time. And so when I initially saw him, he presented on MRI with this, you can see this image that looks like a tooth in his mid-hindbrain. It's called a molar tooth sign because it looks like a molar tooth. And he presented with low tone and developmental delay and lots of recurrent infections. He was in and out of hospital a lot. And in 2013, his younger brother was born and he passed away from this condition, unfortunately. And so we had him in the research study for a super long time, and he had exome sequencing, he had genome sequencing, and he had, eventually, he needed to have RNA sequencing. So in the gene CEP120, which is known to cause Joubert syndrome, we had one interesting variant, but we could not find the other variant. And so we had to use RNA sequencing in RNA extracted from blood from Austin and from a cell line from his brother to show, just showing here the affected abnormal splicing. And so they have a very deep intronic deletion inherited from mom of 57 bases, which causes some intron retention and introduction of a premature stop codon in SEP120 as the second mutation in this recessive condition. And the other one was a missense mutation inherited from his dad. But without those new technologies, both the RNA-seq to know where to look and the genome sequencing then to be able to sequence across that and see that there is an impact, we would not have been able to solve him. And this is Austin here with his brothers and sisters. And he is rare amongst the rare, as Austin's dad says, and he's quite a great advocate for rare disease now, Austin. And the last thing I want to share with you is that when I started here in Ottawa in 2005, I practiced in neurogenetics, and I saw a huge number of families who have a French-Canadian background, but living in Ontario, so not Quebec. Who all had adult onset, slowly progressive ataxia. And over the years, I've seen 11 families who have probably over 60 affected family members. And they present in their 30s, 40s, or 50s with a slowly progressive issues with their balance and coordination, some eye findings, and their back of their brain cerebellum in charge of the balance starts to get a little bit thin as they get older. I've been looking for this gene for 15 years. We did exome sequencing, the earliest versions of it, subsequent versions of it when it was better. We did short read genome sequencing, we did RNA sequencing, we could not find it. And our hypothesis was that all these families had the exact same ataxia, could not prove it. Then we finally had a break. And so collaborating and this group, this study has been led by Bernard Bray, who's a neurologist from McGill, and long read sequencing, as well as some really interesting approaches to try to look for repeat expansions in short read sequencing, flagged that the gene FGF14 has a repeat expansion within an intron. So you can imagine how hard that is to both detect and read, but long read sequencing can see it. So this is an example of long read sequencing here for a couple of these individuals from these families. You can see this is the normal and this is the expanded track. And so you get through the long read sequencing, which delivers chunks of intact data from 5 to 10 kb in length, can read right across that expansion and read it completely. Whereas in short read sequencing, it's all chunked up. And so you need to have some special and innovative approaches to try to identify these repeat expansions, which probably exist all over the genome and are actually pretty hard to identify. So long read sequencing identified this repeat expansion in all of these families. All 11 of them are not now diagnosed with this particular condition, which is actually prevalent in all populations, but particularly prevalent in the French-Canadian population. So there's also lots of families in Quebec that Bernard Bray has identified who have this condition. So when we think about the unsolved families, I told you that we had over 3,000 enrolled in the pipeline, and I just want to talk about a chunk of 1,326 of them for these last couple of slides, and that have gone all the way through part of the pipeline. So this group, 1,326, where we've reanalyzed existing data for all 1,326 families. So that's the exome data they already had as part of either research or clinical care. We were able to make a diagnosis on initially negative data in 32% of families. And that's why when I say, you don't want to throw out the data and just start over, because there's so much discovery left in that data that already exists. In this data, about 9% of the diagnoses were changes in known disease genes that were identified usually in the interim between the last data analysis. 8% of these were novel gene discoveries or new disease gene relationships, like the ones I showed you for Justin and the family with the bone dysplasia. That's about 8%. 2% ended up being diagnosed by a secondary assay, and 13% still have candidate genes that are participating in matchmaker exchange, and we are collaborating with other groups to bring those into the first publications. And then this is where we're at right now in terms of the families that remain undiagnosed after reanalysis. We have had 154 go all the way through the pipeline. So we can evaluate, you start at the top and go through some of those strategies like we talked about with the 4 different types of clinical conditions. How many do we diagnose? And so in that case, about 37%. Now, this data is a little bit skewed because, for example, in the short read genome sequencing, we do have a big cohort of families who all have the same thing, same within the deep sequencing. So it overestimates still, but it will balance with time. But you can see that most of the time in both the deep sequencing and the short read sequencing and the long read sequencing, we are actually finding mechanisms that in known disease genes that we knew about from before, but the mechanism was hiding most of the time. And the long read sequencing seems to be really good at identifying the novel, repeat expansions, for example, that might be the cause. All right, so in the end, where we think we sit is that when we're trying to look at this group of rare genetic diseases, probably the clinical exome sequencing will diagnose about 30%. Reanalysis a few years later will get you another 10%, and novel gene discovery anywhere from maybe 8 to 15%, I think. I think 20% is getting higher, so it's probably tapering off. And then you have these other, alterations, regulations, mosaicism type activities, type of mechanisms, I should say, that need the other omics applied in a logical order to try to solve those. And then there'll be that group that really is complex inheritance, even though they appear to have a single gene etiology. And it really will probably take us, I think, another 5 years to really be able to hammer down this relationship. And as time goes on, the novel gene discovery will decrease, and it'll get picked up into the clinical genome-wide sequencing and part of clinical care. And the last slide I want to show you, just before we have some time for questions, is it's really important to make sure we keep in mind the post-diagnostic odyssey, which I've not talked about a lot today. But once the diagnosis is made for these families, they go into a different odyssey. And we do have some activity in these various areas. But, for example, RDMM is helping when there's no one researching their rare disease. And some of those have gone into some screening for treatments, for example. We have couple of nice examples of that. But the other things around, your clinical care post-diagnosis with respect to clinical care guidelines, expertise, coordinated care, access to treatments if they exist, all of those things are particularly challenging and become the next set of challenges for these families. And with that, I would just like to acknowledge the hundreds of individuals that have participated in our networks over the last 10 or 12 years, all of which I'm nicely summarizing the work of the many with this presentation, and also all of our funders that have supported the various networks I've presented to you today: Care for Rare, the Rare Disease Models and Mechanisms Network, and All for One, that are helping us sort of try to improve the diagnosis and the research around rare diseases for our families that we're looking after here in Canada. So thank you.
Thank you very much, Kym. The lecture was great, and I think it faces us with a challenge that is to make this kind of investigation available to every patient with rare disease worldwide. So the first question is related to that. We have a question about Africa, right? What is the fate of rare disease diagnosis in Africa? If you could comment a little on that.
Yeah, sure. I think the challenges around some of the lived experiences of patients with rare genetic diseases living in countries who potentially don't have access to the kind of healthcare systems, from, for example, where I'm from, it is a big one. And I think that the way some of us have been thinking about that in sense of, how do we approach this globally, is really around making sure that those countries who have interest in moving rare disease, both diagnosis and treatment forward, that we provide mentorship for that and sharing of all the infrastructure that already exists. It would be a shame to think that these folks would end up having to reinvent wheels, etc. Like, it really needs to be more of a global outreach program where they can pick up things that have already been tried and tested and proved and are cost effective, and even better when they're freely available from other groups that have been doing this for a while. And I think that's one strategy I'm particularly interested in for places, countries in Africa, but also many other places around the world.
Okay, thank you. If you read the chat, you see that many people are saying that the lecture was great. And the second question is about the collaboration. It's from your lecture. It's also obvious that many people are necessary to generate this kind of data. Really, it's not only one professional, but if we think in terms of community, how could we build collaborations in this field and build partnership with clinicians? I think the gene matchers things, this is a good example for that, but we'd like to hear your opinion.
Yeah, so so there's a couple of ways. So in terms of building collaboration, so I think building collaborations, as you could see through the Matchmaker Exchange, for clinical groups and researchers, scientists that are working on those genes, has worked really well over the last 7 years. And I've been involved in lots and lots of collaborations, which have all gone amazingly well. I think our approach to the Rare Disease Models and Mechanisms Network, though, probably deserves a pause and a bit of an applaud for that, because I'm very proud of it. It was originally envisioned by Phil Heater, who's a yeast biology at the University of British Columbia, and he and I and others have led that for the last 8 years or so. That has now been was picked up in Europe, Australia, and Japan, and we actually have a rare disease RDMM international group to help us make connections between genes of interest in a country and the best scientists, no matter what country they live in. And so we're working on those sorts of things. So I think some of this, these efforts to build like infrastructure, like we talked about with RDMM, is a great way. And the other one in the US that is getting a lot of traction is something called Model Matcher. And Model Matcher links to RDMM, but it's really about Model Matcher doesn't necessarily come with money. All the RDMMs have grants that also fund the scientists to do the initial pilot experiments. But Model Matcher is actually just about making connections. It's a matchmaker between clinical groups interested in genes and scientists who are interested and have the ability to model those things. So I think things like that is probably one of the excellent ways to proceed.
Okay. Now we have a question about career as a medical geneticist, and maybe since tomorrow is the International Women's Days, maybe we could tell us how you approach to get your career in this field, because the question is related. I know that it changes from country to country, but how will someone will be able to work on medical genetics only if he has a formation on genetic counseling, or if a person has a PhD in genetics, he or she could work on this field. What do you suggest for people interested in doing some kind of work like you were
with families and stuff? Yeah, so my career was, I started out as a researcher, actually. So I started out doing my PhD and decided when I was finished that medicine would be a good way to go, so I could actually see the families that I want to study in the lab. And that's how I ended up going down my crazy road. And in the end, I just wanted the best of both worlds, essentially. And in Canada, we train medical geneticists through a 5-year program, but there are lots of ways to train in medical genetics, depending on where you're from, including training in a different specialty and then doing a fellowship in genetics afterwards. We also train genetic counselors in Canada who practice in our clinics with us, as well as lots of other specialists in genomics and informatics, et cetera, that are all part of these big teams and have various roles to play in various different training programs. So I think if I was to say, how do you get into it? Really, it's about identifying, the area that you're interested in, and then identifying PIs or mentors who can help you get to where you want to go and give you whatever experience you next need in your education to help you get there.
Okay. Now, maybe a shorter question. When you recommend that the reanalysis of genetic data occur?
Very good question. I think we're probably going to land at anywhere between 2 and 3 years. It is a very good question, and one we are trying to answer very systematically here. A lot of the commercial labs in the US offer it once at any time, and most people pick it up after a year or so. But when you're in a single payer system, you need to show the cost effectiveness of that before that kind of decision is funded, because when you do a reanalysis, you do need your scientists who are doing it to be funded to do that. And so I think probably the highest yield would be between 2 and 3 years, unless there's a compelling reason to analyze it earlier. Like, for example, the phenotype has changed dramatically, or there is a pregnancy where this information may impact that pregnancy.
Okay. Now a question about Colton's case.
Yeah, I see it.
They are asking why you could not detect the large deletion.
Yeah, I think that if we probably redid that exome now, we should probably be able to see the 3 exon deletion. But at the time his exome data was produced, the informatics pipelines that we were using were not particularly good at picking up small indels.
Okay. And now, how are multiple SNPs distinguished versus mutations? And this is the question.
How are SNPs? I assume by so 2 things, I guess, if I'm hearing that right, Ida, is that when we're filtering the data, we filter using Nomad, and we filter using our Canadian data set. And so when you're seeing somebody's, massive amount of data in front of you, Nomad will take out hundreds and hundreds of variants and decide whether or not they're rare. And our own data, which is unique to the Canadian population, takes out the other half. And so with those 2 data sets, we usually come down to, a few hundred, depending on the way it's been set up, a few hundred variants to look at. In all circumstances where we can, we always run multiple family members. And so our clinical testing and our research testing is funded for trios or quads or whatever, because the more family information you can have, the less variants you need to look at to decide what's important and what's not.
Okay. And between all the causative genes that you found, some have turned out to be non-coding RNAs.
Yeah, no, they haven't actually. So we have not had any great examples of non-coding RNAs. Now, it may be that we're missing them. That's possible. But we have not had any of those yet.
Okay. So SAB working would like to hear more about animal models of how can we use animal models for discovering of novel gene causing diseases. What do you think?
So your thoughts on animal models of novel defect causing genes. So the way we've been doing it in Canada is to we have a wonderful scientific advisory committee that really looks at and provides the great input that people like myself who are not a model organism scientists to identify which is going to be the best model for that particular disease, and that because of the nature of the gene, the protein, or the mechanism, and they really or the phenotype. So they really give some really thoughtful expertise on, should this be a mouse? Is C. elegans going to be great for this? Should it be Drosophila because it's, spasticity and we can use a walking assay or a climbing assay, for example. So they really help people like myself who don't necessarily know what might be the best model identify what would it be because of the question that we clinically have. And sometimes when we're thinking about treatments too, they can sort of advise and then find us a match for somebody who might be able to have the expertise to develop like a screening assay, for example, and say zebrafish and epilepsy or something like that, which we've done quite a few of those.
Okay, there is another question about animal models. If we take in account genetic diversity, because we know that the genetic background can influence the phenotypes.
Yes, we definitely do that. And when the scientific advisory committee is thinking about which model might be good, we always have the information from our bioinformatics core about the conservation of the gene or the gene family in a particular model organism before we decide which may be best. And then, of course, when background has an influence, the scientists that then proposing the methodology back to our scientific advisory committee will have to have put into their 2-pager proposal how they want to manage that.
Okay, and there are 2 questions here about non-coding regions. If we talked about their RNAs, but you have identified any mutations like in enhancers and things? Like that.
Yeah, no enhancers, but definitely deep intronic splice is probably the biggest one that we've found so far. And we've found some of those using long read sequencing actually. And they're often just large deletions quite deep within an intron. But then when we do the subsequent studies, we see that they have an effect. Enhancers is going to be a fascinating area to go into. And I kind of have this hypothesis that mutations and enhancers may give us different types of diseases than the ones we're currently studying. You can appreciate that because the rare diseases we're studying right now are quite severe, right? They have malformations, they have developmental delay, intellectual disability. And I think a lot of that is impacted by the protein coding genome. But as we start to expand the types of diseases that we're studying, I think that's where enhancers and complex regulation type changes in the genome will start to really say that they're very important.
You know that here we have another question, and I think it's the last question. It's about Africa, but here I'm speaking from Latin America, right? And also countries and population from Latin America, they are underrepresented in the large genomic or genetic banks like GNOMAD, right?
Yeah.
And we know that we have also some very poor countries here in Latin America, maybe with the same situation that we find in Africa. So the question here is, of course, many patients with rare diseases, they are not diagnosed here or in Africa or in other poor countries because of the lack of resources and expertise. Are there opportunities for or training programs for those interested in your research?
Yeah, that's a great question, and one we've talked about quite a bit. So we have a plan in motion. We don't have any training programs like that are funded at the moment. We've definitely taken visiting scientists many times who can come on their own, but what we're trying to develop is actually an outreach funding program to bring people in for real life experience with us in Canada to see how we operate, and then take those knowledge, that new knowledge back home to their own environments. But we're waiting to see if it's actually funded.
Okay.
If it is, we'll advertise on your lovely site.
Okay. So I think now we do not have any other question. I think the session was very interesting. And before I finish, I'd like to know if you'd like to say your last words, Kym, to us.
No, thank you, Ida. Yes, it was a pleasure I very much thank you for the opportunity too, by the way. I want to make sure I say that.
Again, I thank you very much for accepting our invitation, and also to all the attendees, and see you again in the next section. Bye.